Well, if everyone's had plenty of lunch and something to eat and drink, then we'll restart. We've got hopefully a fascinating afternoon for you with the sort of very different themes throughout the afternoon. I'm very sorry to have to inform you that New Zealand did beat England, but it was only by 3 points, so it was close. That's pretty good for us. Anyway, so I'm delighted to introduce Dr. William Pugh. Bill is a basic science researcher based here at Boston Children's Hospital and also a clinical cardiologist. And he's been very much at the sharp edge of understanding how we can use stem cells, human stem cells, to understand the conditions that we're dealing with, and also to potentially offer new treatments and to really personalize to some degree the medical therapy that we can offer for individuals. So it's a great pleasure to welcome Bill Pugh.
Thank you. So this is going to be hopefully a light session that you can enjoy after your meal. This is stuff that I think maybe can give you a taste of what to look forward to in the future, but I think all of this is still in the lab and not really being directly applied to patients yet. I was asked to talk about how stem cells could be used for inherited arrhythmias. And I'm sure you've all heard a lot about stem cells in the news and how they could be used for treating all different kinds of conditions. So as you've heard already this morning, the heart is coordinated by electrical activity, which both permits efficient heart function and also helps to prevent abnormal heart rhythms. And these electrical signals are called action potentials. And I think Vasilios led you through all the different channels that need to be synchronized in order to build a normal action potential, and how abnormalities of one of these channels could cause something like long QT syndrome. So here's, does this work? Okay, so here, for example, this is a diseased heart muscle cell that has a mutation in this channel, and what it's showing is that as a result of this mutation, the action potential became longer, which makes the heart vulnerable to arrhythmias such as torsade. So for a long time, people have wanted to study this in human heart muscle cells. So this is, in an ideal situation, we could take a patient that has a disease like long QT syndrome, and if the patient was really nice to us, we might be able to get some heart cells from the patient. And then we could use those heart cells to understand the disease process and develop new treatments, or maybe use them to predict risk level, or maybe even individualize therapy. But I think you can see the limitations here. It's hard to get people to give us some heart muscle cells. It's not something that we have people lining up to do. And even if we were able to get some heart muscle cells, a property of heart muscle is it doesn't divide, which means you can only keep it alive in culture for a few days at the most. So this really was a huge barrier to all heart muscle research until only a few years ago. And people tried some, of course, not all research stopped. There was still some very productive stuff that was done. One thing that you can do is you can take one of these proteins from a heart muscle cell and just put it into a cell that we can keep alive in culture. Then we can study that protein, but you can see the problem is it doesn't have all of its friends here, so that you can't study how it synchronizes with all the other proteins. Another thing that many people have done is to make different animal models. I think you heard about a mouse model and a zebrafish model, but a big limitation there is obviously that mice are not people. And we've cured a lot of mice of a lot of diseases, but it often doesn't translate into people. So a really revolutionary discovery was made in 2007. And this was just a real breakthrough in which a Japanese researcher named Yamanaka discovered that he could turn a skin cell, or most cells in your body, and you can reprogram them to become a stem cell. Now, by stem cell, what we mean is that cell can then turn into any other cell that we try to force it into. And this was such a revolutionary discovery that within 5 years, he won the Nobel Prize for this. And it's really transformed the way that we do heart muscle research as well as research in pretty much all areas of medicine. So the idea here is you can take now, instead of going to the patient's heart muscle, you take a piece of skin or blood or even urine or a swab from someone's mouth. You reprogram those cells and turn them into stem cells. And one nice thing about stem cells is you can culture them indefinitely, so it's no longer a limited resource. You can then take those heart muscle cells and those stem cells and turn them into heart muscle cells. So now you have heart muscle cells that originated from a patient. So then we can use the patient-specific heart muscle cells to study their disease. And I'll give you an example of some research that my lab has done that's not directly related to arrhythmias, but it is related to heart muscle disease. And this is a rare disease called Barth syndrome, which is a mitochondrial disease that causes a weak heart muscle. And it's due to a mutation in a gene called tafazin. So what we did is we obtained some skin from a patient with Barth syndrome. We made stem cells from the patient's skin. We then turned the stem cells into heart muscle cells. And here, this is some of the heart muscle cells that we put on a plastic dish. And you can see that what was formerly skin is now a beating heart muscle. And this is from a normal person, not from the Barth syndrome patient. So then what we do is we can actually use bioengineering to assemble these cells into heart muscle tissue. So not just random cells scattered on a dish, but cells that are organized into a muscle tissue. So here you can see the muscle cells are all lined up like they would be in a normal heart muscle. And these cells are assembled on a thin plastic sheet so that when you put the cells on the sheet, they actually beat. And by the amount that they bend the sheet, we can measure how much force the muscle is generating. So this is what a normal patient's, we call these engineered heart tissues or heart on a chip. So a normal patient's heart on a chip and the Barth syndrome heart on a chip. And I think you can really easily see that these guys are beating, but these guys are not beating. So we think we made a tissue model of this type of weak heart muscle. So now that we made this model, what can we do with it? One thing we showed is that if we just put the mutant gene back, that we can restore the function of the heart muscle. So of course this makes it attractive to think of whether gene therapy would be useful in this disease. And that's something that we've pursued more and actually shown in, again, we can easily cure mice with this disease by putting this gene back. We also studied the mechanisms of the disease, and we discovered that something called reactive oxygen species is important for this, the cause of this disease. And we showed that if we block the reactive oxygen species with some drugs, that we can turn the weak heart muscle into normally beating heart muscle. So we think that this identified a new direction to go for treatment of this disease. So now let's turn to arrhythmias. So how could you use a in vitro personalized model of an arrhythmia? So one obvious way would be for risk stratification. And here's two examples of risk stratification. You can take a gene variant that you don't know how important it is and then put it in a model and see if it really causes arrhythmia in your model. And the second would be to determine if, we talked about incomplete penetrance, so we could try to predict which patients might have penetrance and which might not. So here's an example. Here's a pedigree. This is just a hypothetical example. Let's say that this patient suddenly died, and genotyping revealed that 3 people in the family have the gene mutation. So a challenge currently is to know what to do with these 2 people. Should they be treated? Should they get a device? How aggressively should they be treated? Now let's just say that the genetic variant in here is what we call a variant of unknown significance, meaning it's not really known if it's a disease variant. It's known that it's different from most people, but we don't know if that means it's going to cause the disease. So this is particularly challenging because now all 3 people share this variant. One of them died, but what should we do with the other 2 people? So one idea might be you could get the iPS cells from these 2 people and compare it, for example, to the siblings and see if these muscle cells behave differently. And now we can actually correct the mutation in these 2 patients and see if it normalizes their activity. So this would give a real way to determine if a variant of unknown significance is disease-causing. This might also help us figure out what causes incomplete disease penetrance. Here's another example of how this might be used for therapy. So one way would be you could use this type of model to discover new therapies, and I think Dave Mylan mentioned that they had used an iPS-based model to test his new compound, and that's an example of how you can use this to discover new therapies. And another way would be to see if you could tailor each patient's therapy to get the optimal therapy. And instead of experimenting on the patient, you experiment on the cells. You find the best therapy for the cells. And then the idea might be that that would be predictive of what would be the best therapy for the patient. And I'll just give you an example that was published from the Gepstein lab. So here again is a normal action potential and then the long QT action potential, and this is from iPS-derived muscle cells. And so this is just what the contractions look like in the normal case, and then in the diseased case you get the action potential, but then you get these delayed after depolarizations, which are the substrate of this arrhythmia. So this group tested some different drugs to see if they could suppress this arrhythmia. So for example, here's the drug nifedipine, which seems to reduce the after depolarizations. Here's 2 more drugs, pinacidil and ranolazine, that also seem to be effective. So for example, you could try to repurpose FDA-approved drugs that are approved for a different purpose and see if they might be effective. Or you might screen hundreds of thousands of new drugs to see if they might have efficacy. So just as a last example, I think this is more in the realm of science fiction. Although much of what I told you just now that is really now possible, just 5 years ago we would have called that science fiction. But this is what, in today's day and age, I would still consider this to be science fiction. So you take a patient's cells, you make their stem cells. We can actually now, this is doable, we can easily take this mutation and fix it. So we have the patient's cells, but then we correct the mutation. So now it's the same patient's cells, all the other genes are the same, except that we fixed the defective gene. But this last step is what we don't know how to do. How can we put these cells back into a patient's heart to restore the normal conduction of the heart? So that step is where we're lacking in information right now. So just as a summary, you know, we've come really a long way in just a short amount of time. I told you this was only discovered in 2007, but it's generated a huge amount of excitement. We think that we can use this technology to stratify risk. Certainly we could do that for variants of unknown significance. Whether we could individualize each patient's risk using this strategy, I think is more uncertain right now. We can definitely use this platform to discover new drugs that might be effective. Whether it could be used to actually personalize each patient's treatment is something that still needs to be evaluated. And as I mentioned in the future, maybe we can use autologous gene-corrected cell therapy, but that's something that is still in people's imagination. So I just hope I've shared with you some of these revolutionary technologies that let us make patient-specific models in a dish. And I hope you can feel the excitement that these advances might bring. And as I mentioned, this field is going incredibly fast. And I think, most of what I talked about, just as I said, just a few years ago, we would have considered impossible. And now it's really possible. And I hope that that rapid advance will soon lead to new treatments for these diseases. Thank you.
Any questions from the floor? Yes, Mrs. Goethe.
I know that in the past, there was a lot of controversy around stem cells, extremely limited, hindered with by using the skin cells to create stem cells. Is that bypassing controversy? What kind of shifting is that?
Well, there's different types of controversies. First, there's ethical controversy over human embryonic stem cells. So, I think we're familiar with the ethical concerns there. So this totally bypasses using any human embryos. This is taking a person's skin and turning it into a stem cell, not an embryo. In terms of, there are a lot of, you may be aware that there have been trials of putting stem cells into patients' hearts. These are patients with heart failure to see if it can make their heart failure less severe. Those trials are, they're controversial in the sense that a lot of patients have been given these cells. I think it's been shown to be safe, but the relative benefit has been marginal.
Simplified the process of creating that induced stem cell? Is it just a matter of shutting off all the genes, turning them all off? So that
there's a magic cocktail of 4 genes that's what that we call them the Yamanaka factors. And there's 4 that will basically turn most cells into a stem cell. And these, so it was only discovered a few years ago, but now I can go and buy a kit to do this from a biotech company. So it's being done pretty much everywhere. It's been simplified to a cookbook.
Well, thanks, Bill, very much for that fantastic oversight.